competition. Science is a big blessing to humanity. In every walk of life, science is there to serve us. Science is also defined as the systematic observation, experiment, and measurement of the nature and behavior of the material and the physical universe, as well as the formulation of the laws to represent these facts in the general terms. In every aspect of our lives, science is omnipresent and omnipotent. Today we have gathered here for the life science experiment competition that is to take place between the students of Gyan Bharati Vidyapith and Gyan Bharati Vidyalaya English Medium. We have among us Mrs. Bhattacharya and Mrs. as judges. Now I would like to call on stage the students of class 7 from Gyan Bharati Vidyalaya English Medium. The students are Ishan Soni, Aryan Rao, Yuvraj Seth, Aman Shaw, Priyanshu Kumar Srivastav, Karthik Sharma. They are going to demonstrate the starch test on the different food items.
I am Aryan Rao from class 7A. Good morning everyone. My name is Aman Shaw from class 7B.
Good morning everyone. My name is Priyanshu Kumar Srivastav from class 7B.
Good morning everyone. My name is Ishan Soni from class 7A.
Good morning everyone. I am Yuvraj Seth from class 7A.
So today we are going to present a life-size experiment of identification of starch on different food samples with the help of iodine. So let us know what is iodine test. The iodine test is a chemical test used to distinguish mono or disaccharides from certain polysaccharides like amylase, dextrine and glycogen. This variation has termed as starch iodine test that is used to indicate the presence of glucose made by plants in the leaves. So let us know what are the materials required for it. Firstly, we have watch glasses and our samples like potato, powdered cashew nuts, bread, a slice of cheese, an iodine solution and a dropper. So let us start with the experiment. So first we will take iodine with the help of dropper and put it on the first sample, which is potato, and let it be for some time. Again we are going to put some drops of glucose iodine into powdered cashew nuts. Let it be for 2 times. Thirdly, we have a food sample of bread. Okay, with the help of dropper, we are going to put some drops of glucose iodine in the bread. And our last sample, which is cheese, again with the help of dropper, we are going to put some glucose iodine on it and let it be for some time. So let us know what are the principles on which this experiment works on. The iodine test is based on the fact that polyiodine ions form color absorption complexes, helical chains of glucose residue of amylase, blue-black, dextrine, black and glycogen reddish brown. Monosaccharides, disaccharides and polysaccharides like cellulose remain colorless. Amylopectin produces an orange-yellow hue. The reagent used in this experiment is glucose iodine, which is an aqueous solution of elemental iodide and potassium iodide. It is observed that the helical structure of the glucose residue is a key to this test. Further, the resulting color depends on the length of glucose chain. So let us see what has been observed in this food sample. Our first sample is potato. We clearly see a black color has been observed in it, so it proves the presence of starch in it. Our next sample, which is powdered cashew nut, we can clearly see that no observed in it, so it results in a negative test of starch. Thirdly, we have bread. We can clearly see a purple color has been observed in it, so it results in a positive test of starch. Fourthly, our slice of cheese. We can clearly see that no change of color has been observed here, so it indicates the negative test of starch. So let us know what are the results of it. A blue, black or purple color represents a positive test of starch, indicating the presence of starch. If there is no change in color, it results in the negative test of starch. Let us know what are the uses. This experiment is used to indicate the presence of starch in some food sample. Similarly, this test is also used to indicate the presence of photosynthesis in plants. So let us know what are the limitations of this test. This test cannot be performed under acidic conditions like starch hydrolysis. Secondly, this test is a qualitative test and doesn't signify the concentration of starch. Thank you and have a nice day everyone.
We are going to show different types of flowers. Flowers either be completed or incomplete. By the name, we can understand that a complete flower has all four parts: calyx, corolla, androecium, and gynoecium. Here, in this hibiscus flower, it is a complete flower. Now, what is an incomplete flower? A flower without one or more parts is called incomplete flower. A tube rose is an example of incomplete flower. The male flower of pumpkin or cucurbita maxima, by dissection we can see that that is only stamen, calyx and corolla. No pistil is there. But in the female flower of pumpkin, there is calyx, corolla and pistil. No stamen is there. So these are unisexual and incomplete flowers. Now on the basis of the position of ovary, the flowers also have about three types: epigynous, perigynous, and hypogynous. In epigynous flower, the ovary is positioned inferior to calyx, corolla, and androecium, and the thalamus encloses the ovary inside it, and all three parts are below it. In this pumpkin flower, we can see that perigynous. In perigynous flower, the ovary is at center and all the four parts are in same level. In this rose flower, we can see that hypogynous. In hypogynous flower, the ovary is seen to be superior to all parts, and the thalamus is this hibiscus flower. You will see that the calyx is at the below and the calyx is at the below and all the three parts are in below, and the ovary is superior. Thank you.
I am Aishwarya of class 7B. We know that flowers are the most beautiful creature of nature. But actually, what is flower? Flower is a male reproductive part of a flower. They are not only taking part in reproduction, they are also a storage of food in the form of nectar for some living organism. Now, we are going to see the basic structure of a complete flower. Can anyone identify this flower? Yes, this is China rose, hibiscus or java flower. Now we are going to see the structure of flowers. There are five petals. Now, this is an androecium or stamen. This is androecium or stamen. This is the main reproductive part of a plant, consists of anther and filament. Anther is a yellow sac-like structure involved in producing pollen grains. The upper part of the flower is called stigma, and the innermost part of the flower is called ovary. Now, we are going to see the pollen grains under the microscope. Pollen grain is a reproductive part of the unit of the flower or the plant.
Good morning to all judges, teachers and everybody present here. Today, we, the students of class 8 of Gyan Bharati Vidyalaya English Medium, are going to present before you a life science experiment on pollen germination. The participants are Ayush Singh,
Ashutosh Agarwal,
Akshay,
Pratham Gupta.
Now, my friend Ashutosh Agarwal will introduce you about the pollen grains.
So let us know what are pollen grains. Pollen grains are microscopic structures which Pollen grains are microscopic structures
of landing of pollen on the stigma, the pollen tube growth, and the entry of germ cells are the events covered in the pollen germination. There are several factors which affect the pollen germination. First, pollen viability. Fresh, mature pollens from healthy flower are most likely to germinate. Second, temperature and humidity. Warm and moderate climate provides optimal condition for the pollen tube growth. Third, nutrient availability. The sugar solution provides essential energy for the pollen germination. Now my friend Akshay will continue about.
Good morning. The first step involves the necessary materials are fresh hibiscus flower, a beaker, sugar solution, filter paper, microscope, and cover slip. The first step involves preparation of a nutrient solution. Here we have taken sugar thoroughly dissolved in water. It is few drops of this sugar solution is added on the top of the slide. Then brush or finger is used to gently dust few pollen grains from the stigma from the stamen of fresh hibiscus flower. To the center of the slide where sugar solution is present. Then a sterile cover slip is gently placed on the center of the slide, avoiding any air bubble to form. Excess sugar solution from the edge of the cover slip is soaked with a filter paper. Then the slide is kept undisturbed for 30 to 45 minutes. Since it's a time-consuming experiment, we have prepared the sugar solution earlier and the pollen grains from fresh hibiscus flower is submerged. Finally, the slide is ready to be observed under a microscope. submerged in nutrient-rich solution. This is characterized by enlargement of tube cell. The germ pore eventually forms the pollen tube. The generative cell nucleus produces a pollen tube and two male gametes. The male gametes are either spherical or lenticular in outline. The precautions we have to take during the experiment. Fresh hibiscus flower should be necessary for this experiment, and the cover slip, between the cover slip and the slide, air bubbles are avoided. Thank you. Have a nice day.
Teachers, judges, and my friends, I am Rishabh Agarwal of class 8A of Gyan Bharati Vidyapith. Can you tell me what is this? Can you tell me what is this? Yes, it is a leaf. Today I am going to discuss the outer and inner structure of a leaf with the help of the cross section under microscope. Leaves are thin, flat organs responsible for producing photosynthesis in the plants. This part of a leaf is thick and is known as a petiole. This part is known as petiole. It is very thick and bright in color. This part of a leaf, when the leaf is meet in the stem, this is known as the leaf base. And the central part surrounding the leaf is known as the leaf blade or lamina. It consists veins and windlets. Now we are going to see the internal structure of leaf. For that, we have to cut the leaf with the help of blade, and then we have to observe it under microscope. My friend Abhay will do it. The outermost epidermis layer is present. Below that, you can also see photosynthetic mesophyll tissue is also present. We can also observe vascular tissue, xylem and phloem tissue is also present for gaseous exchange. The central part within the leaf is called nucleus. Surrounding the nucleus is the nucleolus. Thank you.
I am Skandar of class 8B from Gyan Bharati Vidyapith. Today, by the help of onion cell structure under microscope, I am going to explain what is a plant cell. My friend Rohit Yadav is making ready the structure until I am explaining you what is a cell. A cell is the unit of life of an organism. An organism is made up of a cell. Inside the cell wall, there was a semi-solid liquid thing, that is cytoplasm. We can also see the nucleus. A plant cell also consists chloroplastic, endoplasmic reticulum, vacuole, ribosome, and etc. and mitochondria. Onion cell does not consist mitochondria as onion is the underground modified stem. Thank you.
Good afternoon everyone. Good afternoon to the students of Gyan Bharati Vidyapith and Gyan Bharati Vidyalaya English Medium. I am here with the results. In start taste on different food items, the winners are Gyan Bharati Vidyapith. They have flowers and pollen grains. And in the class 8 section, the winners are Gyan Bharati Vidyalaya